Welcome to the lecture 37 of Introduction to Complex Biological Systems. Today I am going to talk about mechanisms of evolution. So in this week I am discussing evolution, the history of life. So I started discussing protein evolution, right? So, now today I am going to talk about the major molecular basis of protein evolution. So what is this? The change in a protein sequence or structure over time, that will be protein evolution. And the molecular basis of this protein evolution is mutation. Now remember that this mutation happens at the DNA level and the change is reflected in the amino acid level. So if the change in DNA results in change in amino acid, then you have a mutation in the protein. So this mutation can be a point mutation where only a single nucleotide is mutated to something else. It can be insertion or deletion. So meaning a big chunk of sequence is inserted between a DNA sequence. So a small chunk is inserted or small chunk is deleted. Gene duplication. So the same gene is copied again so that you have two copies of the same gene, and then one of the copies gets mutated because it is not behind the correct promoter, and then it becomes a different protein, maybe with related function or maybe with completely different function. And then there are gene fusion and fission. So two genes can get combined to form a chimeric protein with completely new function, or a single gene can be split into two different genes so that they will have different functions. So these are the basic mechanisms by which protein evolution happens. So since the evolution happens or these mutations occur in the genetic level, we have to look at the genetic table or the genetic code. Here each amino acid, the 3-letter code for each amino acid is given, and it is important to pay some attention to this genetic code. Methionine has only one codon. Along with methionine, tryptophan is the only other amino acid which has only one codon. So methionine and tryptophan, these are the only two amino acids which have only one codon dedicated to them. This means that any mutation, any single mutation on methionine or tryptophan will result in a change in amino acid. For example, AUG codes for methionine. If G changes to A, it will code for isoleucine. If G changes to U, it will code for valine, right? So on and so forth. If this U changes to C, then it will code for threonine. If this A changes to C, then it will code for leucine. So change in any one of these three nucleotides will result in a mutation because methionine has only one codon dedicated to it. On the other hand, if you see valine, valine has 4 codons dedicated to it, which means that any change in the third base, which is the vowel base, will not result in any mutation. So if this U changes to C or A or G and vice versa, it will still remain valine. Right. So it turns out that there are amino acids, for example, isoleucine has 3, there are many amino acids which have 2, and there are many amino acids which have even up to 6. For example, leucine, it has 6 codons dedicated to it, and arginine, so 4 here and 2 here, they have 6 codons dedicated to them, right. Another important aspect of this genetic codon or this table is that you will see that similar type of amino acids tend to be in the nearby places. For example, if you look at aspartic acid and glutamic acid. So if let's say aspartic acid is encoded by GAC, if this C changes to U, it will remain as aspartic acid, so no change in amino acid. However, if this C changes to A, then it will become glutamic acid. Now both have acidic side chains, so functionally it might not be a very bad mutation, right. Similarly, if it changes to G, then again it becomes glutamic acid. So a negatively charged residue remains a negatively charged residue. So these are similar residues. And this genetic code or the plan or the design of this genetic code determines what type of mutations you will see more often and what type of mutations you will see less often. For example, if you want to mutate a glycine to, let's say, proline, then you will have to change at least 2 nucleotide. If you want to change glycine to serine, then you will have to most probably change, so again 2 nucleotide, right. So mutating 2 nucleotide, the chances, 2 consecutive nucleotide, the chances are much less compared to a point mutation. So here is a summary of what I just discussed. In genetic code, there are 3 bases per codon, right. So each amino acid is encoded by a codon, and in one codon there are 3 bases. Genetic code is nearly universal. It is not absolutely universal, but it is nearly universal among all organisms. 4 types of bases: A, T, G and C. So if you consider RNA, then it will be A, U, G and C. Out of the 64 possible codons, 61 code for 20 amino acids, 3 are stop codons. Highly degenerate codon, only tryptophan and methionine have 1 codons, right. Some amino acids such as leucine, arginine, serine, they have up to 6 codons. I said isoleucine has 3 codons, and then there are other amino acids which have 2 and 4 codons, right. So it is a highly degenerate code. Vowel base, which is the third base of a codon, it is often degenerate. So for example, we saw the example of serine and glycine. If you change the third base, it will not result in any change of the amino acid. Silent mutations. So if a codon is changed into another codon, but it does not result in a change in the amino acid, then that will be a silent mutation. Now if we consider point mutation, then there are 9 possible point mutations. So let's take the example of glycine. So let's say glycine, it is encoded by, so in a particular protein, let's say the codon that we are looking for, here the glycine is coded by GGA, right. So if you consider GGA, and if I mutate this third position, I can change it to G or C or T. Now GGA codes for glycine, but if I change this third base or the vowel base to G, C or T, it will still remain glycine. So these are all silent mutations. Instead of this, if I mutate the second base, so this G becomes C, A or T, then GCA will code for alanine, GAA will code for glutamic acid, GTA will code for valine. If I mutate the first one, then this will code for arginine and stop codon. So these are the 9 possible mutations for this particular codon, which is GGA. It turns out that 3 of them are silent mutation. Glycine to alanine might not be a very drastic mutation, so the protein might be able to tolerate that, because alanine is the next smallest amino acid after glycine. Glutamic acid might be a little problematic because we are putting a negative charge. So if the glycine is something that is buried, then this will result in denaturing of the protein. Glycine to valine, again, if the glycine is buried, then we are putting these extra carbons, and that can create problems. Glycine to arginine, smallest residue to one of the largest amino acids, again if it is somewhere buried, then this will create problem. And glycine to stop codon, so this will result in a truncated protein. So for example, let's say if my protein is 150 amino acid long, and if this glycine is in the position 30, then this will be a stop codon. So only first 29 amino acids will be encoded, remaining will not be synthesized. So it will be a small peptide of only 29 amino acids. So it will be a non-functional peptide. So this is a drastic mutation. This will definitely cause problems. Depending on where the glycine is, if it is in a loop, then any of these mutations might be tolerated. If it is something that is buried, then most of these mutations might not be tolerated. So all of these factors will determine whether this particular mutation will be acceptable or detrimental to the structure and function of a protein. So point mutations, 9 possible point mutations per codon are possible. So since there are 61 codons, 61 times 9, 549 possible mutations are possible. Okay. So 549 point mutations are possible. 25% of this, that is 134, are silent mutation, which means that change in the nucleotide will not change in the amino acid. That is because of the degeneracy of the codons. 24% are conservative mutations, which means that the amino acid that it is mutated to will have very similar physicochemical properties. For example, arginine gets mutated to a lysine, so both are basic, or aspartic acid gets mutated to glutamic acid, or phenylalanine gets mutated to tyrosine or tryptophan, right. So 24% are conservative. Okay. So you will see that almost 50% of the mutations are most probably not going to create much problem. 18% are moderate. So moderate means, for example, serine to alanine. Okay. So this serine side chain OH is polar. It can form hydrogen bond, which will be absent in case of alanine, but again both are small side chains. So this should not be a very big problem. 29% mutations will be drastic. So these 29% mutations can create big problems. For example, glutamic acid to arginine, where you are completely changing the charge on the side chain. 4% will result in stop codon, as we saw in case of the glycine. So this will be a very bad mutation, because if there is a stop codon, then the protein will become a truncated protein. So it turns out that majority of these mutations are not going to be a problem, right. So there is a 50-50 chance of resulting no major perturbation, because 25 plus 24, that is 49%. So almost 50% of the proteins will not create major problems. Now if we think about amino acids, there are 190 interchanges that are possible, right. So you have 20 amino acids. Each amino acid can be mutated to 19 other amino acids, and alanine to leucine or leucine to alanine, we can call this the same interchange. So you divide it by 2, so that gives us 190 interchanges. 75 of these interchanges can be achieved by single substitution. So these 75 interchanges will be seen more often, because a single base substitution is more likely to happen. Okay. 101 changes by 2 base substitution. So this will be less likely, and 14 changes where you need to mutate all 3 bases of the codon. So this is the least likely interchange that you will see. So this inherent bias in the genetic code and the fitness of mutation in the context of the protein determine the frequency of amino acid substitution observed in history of evolution. So this is something, this frequency is something that I have discussed in the previous lecture. Okay. So we have already seen this, right, in the previous lecture, that there are certain amino acids depending on the protein. Some amino acids will be absolutely conserved. Some amino acids which are in this loop, they can be changed to anything, whereas some amino acids which are in the core will be highly conserved, right. So again, for globin, which had 150 amino acids, only 2 are absolutely conserved. These are the histidine and the phenylalanine. So what are the general features of amino acid substitution? Hydrophobic residues, which are typically inside the core of the protein, and hydrophilic are on the surface of the protein. So side chain packing is as dense as crystals of organic molecules. So this packing of the side chains in the core of the protein, they are very dense. So there is no gap in the core of the protein, which means that mutating those amino acids which are in the core of the protein will be difficult, right, because whatever mutation you make, you have to compensate that change with some other mutation, whereas charged side chains are on the surface. So charged side chains are rarely found in the interior of the protein. Also, almost all interior polar groups are involved in hydrogen bonds. So these are some of the restrictions that we have for a protein structure. So together this put a lot of restraints on amino acid substitution of residues in the interior of a protein. So proteins which are in the interior, mutating them will be harder than proteins which are on the surface. For example, if I have a protein like this, if there is a arginine or lysine that is sticking out from the surface, let's say this is arginine or lysine, I can mutate it to any other amino acid, for example serine or even aspartic acid, if it is not involved in any salt bridge, right, or if it is not involved in the interaction of any other molecule. So any charge group will be happy on the surface. So surface residues will mutate more frequently compared to the core residues that are present in the interior of a protein. So evolutionary changes in the protein interior are much rarer than changes at the surface. Internal changes tend to compensate each other. So if you mutate one residue and you lose a methyl group, then you will have to mutate another residue where you have to add a methyl group. Then only the packing will be maintained, right? So that is what it is shown here, that isoleucine to valine will result in loss of a methyl group. This has to be accompanied by an adjacent glycine mutation to alanine. So you add a methyl group to glycine to make it alanine, so that the internal packing remains the same. So based on this, what people have observed is these are the frequencies of amino acids. So these are the frequencies of amino acids means that if you consider all different protein sequences and you see that how often alanine shows up, how often glycine shows up, things like that. So you will see that tryptophan and methionine, which have only one codon, their frequency is very less. On the other hand, leucine and serine, which have 6 codons, their frequency is one of the highest, right? One oddball that is here is arginine, because it has 6 codons, however, its frequency is less, not as high as leucine and serine. So the graph that you see here is something that is plotted. So this is the expected frequency that is plotted for these amino acids. This expected frequency is based on how many codons are there and how often those codons are used. So based on that, and on the y-axis we get observed frequency, and you will see that there is a nice correlation between these two, which means that amino acids which have more codons are observed more often than amino acids which have less codons. However, arginine is the exception, and you can see that it is an outlier. Okay. So based on this frequency, the expected frequency, the observed frequency, how many substitutions are happening for a particular pair, whether it is same as observed, same as expected, more than expected, or less than expected, this type of Blossom 62 matrix has been generated. So we saw this, how this matrix is generated in the last lecture. So again, just a brief summary. Numbers which are 0 indicate that this particular substitution, so here 0 is for lysine, so lysine to asparagine. This substitution is almost the same that you would expect. Okay. So the observed frequency is same as the expected frequency. On the other hand, if I go to this, lysine to glycine is less than that is expected. So whatever frequency we expect, the observed frequency is less than that. So we have a negative number. On the other hand, if I go here, lysine to arginine, it is +2, which means that the observed frequency is more than expected. So lysine to arginine or arginine to lysine, this exchange is observed more than you would expect based on the natural frequency of these amino acids. Okay. So this was all about point mutations, right? So apart from that, there can also something that can happen, that is insertion or deletion. Now, I have a gene, so that encodes for a particular protein. If something is inserted, now if this insertion is multiple of 3, then my reading frame is not changed, okay? Which means that if I insert, let's say, whatever my reading frame was, so I insert 6 amino acids, so it means that 2 amino acids will be inserted, right? But if it is not multiple of 3, if I insert 7 amino acids, then the first 3 will code for one amino acid, second 3 will code for another amino acid, then this last one, the 7th, plus 2 more from next will code for a completely different amino acid. So it will change the sequences that will be downstream of this insertion. So this is the same, exactly same for both insertion and deletion. So any multiple of 3 will not cause much problem, but if it is not multiple of 3, then it will create frame shift for the downstream sequence. Gene duplication. So this is something that happens quite often. So a particular gene gets duplicated means that it gets copied again. Now this gene which is there, it has a promoter on its upstream, and that promoter will regulate its expression. But if it gets copied somewhere else, and if there is no promoter which results in its expression, then that extra copy will start getting mutations, right? And by chance it can show up in context of another promoter so that it gets expressed and it will become a different protein. In many cases what happens is that gene duplication happens with the promoter so that all the copies are actually expressed. So this is something that is observed for ribosome and histone. So these proteins have multiple copies of the same gene. There are several advantages of this. One is that your gene dosage is more, so that because you need a lot of ribosome and histone proteins. So since we have more number of genes, more mRNA will be produced and you will get more of this protein production. Second is that if one copy is corrupted by mutation, then the function will not be hampered because you have other functional copies. So in most cases, mutation in the extra copy result in non-functional protein and the gene is eventually lost forever. But in these cases, the gene will still be there because you have extra functional copies. So for example, Saccharomyces cerevisiae diverged from its ancestor by whole genome duplication, which means that this particular yeast, the whole genome was duplicated, and 85% of these new genes were eventually lost, but 15% of the genes resulted in some new protein, and that resulted in the evolution of this new organism. So in some cases, new function evolved resulting in a new protein. So after gene duplication, the extra copy can evolve into a new protein with a new function. So there is a particular term that is used a lot in this context, which is called homology. So homology means proteins that evolve from a common ancestor are homologous proteins, right? So this is a yes or no condition. There is no partial homology. So two proteins are either homologous or they are not homologous, right? However, there are problems that can happen if there are multiple domains in a protein. So one example is when different parts of a protein have different origin. So LDL receptor, it is composed of domains which are homologous to the complement component C9, so 34% homology, and EGF-like domain, so it has 48% homology with this. So it means that some sort of fusion happened. So two different domains came from two different genes and they got fused, and now you have a protein which has homology with two different proteins. Okay. The animal fatty acid synthase comprises of two multifunctional polypeptide chains, each containing 7 discrete functional domains, and each of these domains might have come from different proteins. So these are all examples of gene duplication as well as fusion, right? So multiple genes, they come together to form a single gene. However, they will fold as independent domains. Similarly, gene fission can also happen, that if there is a protein with two different domains and if the gene breaks here, then you will have two different proteins where each domain is now a separate protein. So evolution in most cases is divergent evolution. So what is a divergent evolution? So let's say there is some ancestral species which has a particular protein and that is denoted by P0. So this is the common ancestor. That species can survive, so it remains the same and the protein also remains the same. So let's say we are seeing after, this ancestor originated maybe a billion years back. This is the present time. So the same ancestor is there. So there is no change in the protein. However, a new species can form where this protein is changed slightly. However, it performs the same function in a different organism. So this is called speciation. Okay. So it's the same protein. The sequence has changed, but the function remains the same. The same protein or the same gene can get duplicated. the function changes may be very similar or slightly different, but in the same organism. So, in this case it is differentiation. So, the sequences will be very similar, however, they have slightly different functions, or it can be completely different function, right. So, these two are in the same organism. So, these two proteins are originating from the same ancestor, however, they have gained so many mutations that they can now perform either related function or very different function. So, this will be differentiation. In this case, this sequence will be slightly different from this, but they perform the same function in different organism. So, this will be speciation, right. So, the ancestral protein is altered over time. It yields an array of descendants with same, similar or different functions. So, these are all homologous proteins. So, these all proteins will be homologous to each other and to this ancestor. So, whatever I mentioned is written here. So, P1 there is no change in comparison to the ancestral protein. P1 dash, this is speciation. So, such proteins are called orthologs. So, sequence alignment of orthologs proteins provide important information on their evolutionary history, right, because it is the same protein. So, for example, we saw cytochrome C in all these different organisms, and then we used that to create this tree of life, right. So, these type of orthologs are very useful. Differentiation, so here the protein has related or similar function, but not exactly the same function. So, this requires gene duplication. Examples will be chymotrypsin, trypsin and elastase. These are all enzymes which cleave the peptide bond. So, these are called proteases. They have very similar fold, right. So, chymotrypsin cleaves after this bulky aromatic groups, trypsin cleaves after the basic amino acids, and elastase cleaves after residues which have small hydrophobic side chains, right. So, these are all proteases and they will be this result of differentiation. And then the third one is where the function is completely unrelated. For example, hen lysozyme which breaks the bacterial cell wall, and alpha lactalbumin which is not even an enzyme. Okay. Alpha lactalbumin, it is a protein in bovine milk that regulates lactose biosynthesis. These two proteins are 38% identical and they have similar fold but have completely different functions. So, this was divergent evolution. We also have something which is convergent evolution, and this is something that is rare, and we will see example of convergent evolution in the next lecture where I discuss evolution of I. So, convergent evolution means that two unrelated proteins, they start from different ancestors, but they converge to the same function. Okay. So, they start out with different sequences and they remain different, but they evolve to perform similar function. So, in case of I, we will see that eyes have evolved from different ways and they perform the same function, which is vision, but the designs are completely different. So, for clear cases of convergent evolution, proteins have no recognizable sequence similarity and of course identity. Their structures will also be different, but they will perform similar function. So, at the protein level, a very good example will be that of chymotrypsin and certolysine. So, both are proteases, both are serine proteases. Chymotrypsin has two key beta barrels, Greek key beta barrels. So, that is its structural topology, whereas certolysine has beta parallel beta sheet topology. So, in both cases the active site residues is serine. So, serine is the one which performs the reaction, and both employ a charge relation which involves aspartic acid, histidine and serine. So, the active site has the same residues and the reaction mechanism is also very similar, but the structure is completely different and the sequence is also very different. So, this is the structure of chymotrypsin. It has two domains. You can see there is one domain here, there is the other domain here, and if we look through this domain from this direction, it will look like a beta barrel like this. And the active site residues are in the interface of this beta barrel. So, there is a serine, there is a histidine, this is a aspartic acid, and together they form the catalytic triad. So, I have already discussed chymotrypsin in details in a previous lecture. One important thing that I should point out here is you will see that these two domains look very similar and both have this beta barrel structure. So it is hypothesized that the original chymotrypsin had only one domain, and then gene duplication happened where the same gene was copied just right next to each other to result in this complete protein, and that resulted in this 2 beta barrel structure. However, it is interesting to note that serine is in one of the beta barrels and these two are in the other beta barrel. So the original enzyme was most probably only this, because you absolutely need the serine, and if you mutate these two, you will still have some function. It will be very poor, but it will still have some functionality. So once this gene duplication happened, it became a much better enzyme and got selected for. So this picture makes it more clear. This is a serine and these are the other two residues, and together they form the catalytic triad. So this is something that I have discussed in details in a previous lecture. This is the mechanism at a glance. I have already discussed this in a previous lecture. So serine, chymotrypsin, it is formed as a pre-pro protein, and then it gets activated by other enzymes, and then it forms this functional enzyme. So if we look at certolysine, it looks like this, and you can see its topology is completely different, and also it looks very different in structure. However, it has the same 3 residues in its active site, very similar arrangement and the same residues in the active site. So it means that the same catalytic active site has evolved from 2 different ancestors. So they have converged into the same function. That is why this is an example of a convergent evolution. So this is the active site of the certolysine. So we have a serine here, we have a histidine here, and we have an aspartic acid here, and together they form the catalytic triad for certolysine. So for this lecture, you can go through these books, especially Lehninger Principles of Biochemistry and Molecules of Life, these two. Thank you.